Let's get continue. Okay. Are you with me now? Okay. As I said, the preparation and use of standard curve is important in order to measure any kind of the solid, any kind of the molecules. But we are fortunate. Most of these lab tests, they are measuring and by the analyzer, and the standards are created by the instrument itself. Okay, we are going to use um, um, such plots as graph manually only in a small size of labs or in a research labs. Okay, but in order to understand uh, the the techniques that we are using, maybe um, help us uh, to get the point. Now, okay, note the intervals on the graph papers, okay, in order to plotting a standard curve, and axes must be properly labeled, and you also have to review source of the errors if you uh, notice. And once the standard curve has been plotted, it is used to calculate the concentration of any unknown that were included in the same batch, and this as a standard used to make a graph. Okay, we if we need to measure an amount of glucose in blood sample or cholesterol in the blood sample, we prepare a standard solution with a glucose, different concentration, and the glucose. We measure absorbance of each standards and then plot a graph, okay, with different concentration. It should be straight line. And then we are using this standard plot for a unknown amount of the glucose. Okay, for each method, we have to prepare a standards. Okay, for glucose, for creatinine, for cholesterol, for any kind of the test. And then for each method, we have to have a a graph, okay? And we also need to check them periodically, whether it's working well, okay? Because by the time the standard may be spoiled, okay? Or the reagents, we don't know, we have to check them periodically. Okay, now the instrument used in spectrophotometry or photometry, they base it on the same techniques. And the parts essential to the spectrum photometers, as you see here, light source, okay, and wavelength isolator, a filter. Here, these two, okay, consist of a filter, okay, and wavelength and okay, isolator and cuvettes, okay, and also the electronic measurement devices, okay. These can be either a photoelectric cells, okay, and also plus and galvanometers in order to make a increase its strength. Okay, let's see how does the spectrophotometer work. should be good. Now, okay, calibration of cuvettes, okay, is important. Care and handling of the spectrometer photometer is all important in order to get a high quality of results. Okay, and the quality control test for spectrophotometers. Okay, wavelength accuracy. Straight light is any wavelength outside of the band transmitted, and the linearity is the reportable range of the test system used in the laboratory. Okay, these three are a sign of the quality control test. Okay, wavelength accuracy is checked with standard absorption solution or filters. Okay, um, uh, we have to okay, check standard absorbing solution or filters periodically in order to have a accurate wavelength, and also the linearity of the test, which is also known the reportable range of the test system, is important. Now let's see the spectrum photometers.
In this video, I'll be demonstrating how to use a spectrophotometer. A spectrophotometer is used to read the absorbance of various samples. Now, when you first get your spectrophotometer, you will need to remove the cover and then turn it on. And there's a dial in the lower left-hand corner. Once you turn it on, if you look in the window, it will begin its initialization process, and this can take several minutes. So we're going to wait for this to happen. Once it is initialized, it is time to set the wavelength that you're going to be using and to blank the machine. In order to set the wavelength that you are using, you use these two buttons here that says NM arrow up or NM arrow down. So for this demonstration, I'm going to set the spectrophotometer to 540 nanometers by pressing on the NM down. So now that it is set to 540 nanometers for this example demonstration, I'm going to blank the machine. This will require a solvent blank. All you do is you open the chamber. You put in your solvent blank, which is in a prepared cuvette, close the chamber, and press the button that says 0 ABS 100% T. And when you do, you'll see the message setting blank. And when it's done, it'll read the wavelength that you've set it at, in this case 540 nanometers, and 0.000 A, and that A simply stands for absorbance. In this class, you will only be doing absorbance readings, not percent transmittance readings. So what we've done now is we have canceled out all of the absorption that is due to the solvent that's used in the experiment. So now to read the wavelength of samples, you remove the solvent blank, you put in your sample cuvette, close the chamber and read. In this case it says 0.226 is the absorbance. If I want to read multiple samples all at the same wavelength, I don't have to do any readjustments. I just need to open it up, put in the next sample I want to read, close it, and then read the absorbance for the subsequent samples. You do need to re-blank the machine anytime you change the wavelength. So if you have to change the wavelength, you reset the wavelength, you put in your solvent blank, and once again, press 0 ABS 100% T.
Okay. This is a simple way how to measure a sample via spectrophotometry, but is in most cases is not simple as we see here, because for enzymes you have to measure a absorbance within a period of the times. Let's see, for 1 minute, okay? And it is not simple, but if we get the point how does photometer works, will be enough at these levels. Okay, now the principle and quality control about the measuring of samples via spectrophotometry is other issues. A beam of the light is directed at a flat surface. Okay, this is about a reflectance spectrophotometry. Is different from spectrophotometry and photometry. Okay, as the name implies, reflectance. Okay, I will show you how it does works, how it different from regular spectrophotometer or photometer. Okay, in this case, a beam of the light is directed to a flat surface, and the amount of the light reflected is measured in a reflectance spectrophotometer. The light reflected from surface of colorimetric reactions is used to measure the amount of the unknown color products generated in the reaction. Okay, you reflected the light onto the sample tubes. Okay, you expose the light onto the sample tubes and measure the light amount which reflected from the test tubes. Okay, and now, okay, the part of the reflectance spectrophotometer, a lamp, a source of the light, is similar to the photometer, spectrophotometer, which passes through the filter and a series of sleeves and is focused on the test surface. Okay, some light is absorbed by the filter, and the remaining light is reflected. Now, the reflected light then passes through a series of the slits and lens, and then onto the photodetector devices, where the amount of the light is measured and recorded as a signal. Now, I am going to show here this diagram explain the mechanism of the reflectance spectrophotometry. As I said, this is A is a light source. Okay, then you have different slits and filter. This is a filter, lens. Okay, now you send the light through the slits, through filter, lens, onto a surface, test surface, which is test tubes or cuvette. Okay, and then some of the lights will reflect. Okay, we are going to measure the intensity of the light which reflect from the test tubes. Okay, is come onto the other lens. Okay, and through the slit or lens, and then onto the detector or photodetector device, and read out device will give us a graph of these lights. Okay, there are several routine tests based on the reflectance spectrophotometry, which are most common tests that we are using in our daily life. These are the common point of care testing, okay, and self-testing instrument include for quantitation of the blood glucose in maintaining good diabetic control is based on the reflectance spectrophotometer. Also, chemistry and therapeutic drug monitoring analyzers systems are also employ this technology, the reflectance spectrophotometry. Urine analysis, as you see, these test strips for urine also based on this technology, these techniques. Okay, that means there are lots of tests based on the reflectance spectrophotometer that we are using in the therapeutic drug monitoring, in the chemistry, which we call dry chemistry. As we see here, these are test slides for dry chemistry and also for urine analysis, urine testing. There are various instruments use dry reagent reflectance spectrophotometry. Okay? Or you can also use the same strips manually. Okay, as I said here, this is a kind of the instrument which based on the reflectance spectrophotometer. This is a test slides, okay, dry, which we call dry slide technology, okay. The reagent embedded into these slides, okay, which we need for the measuring of the test, okay, and then the the this the source of the light, we send light onto these slides. Okay, some of them will reflect from the slides. It depends on the amount of the molecule that we are measuring, and the detector will measure the intensity of the light reflected from the test slides, from the test tubes. Okay, we also have a fluorescence spectrophotometry, which is also a kind of the reflectance spectrophotometer. And fluorescent light is used the result of the absorbance of a photon of radiant energy by a molecule. Okay, if molecule release a fluorescent light, then you can use fluorescence spectrophotometer. Okay, when excess energy is ejected as a photon, the result is fluorescent emission, which can be measured. Okay, the intensity of the fluorescence is determined by using a fluorometer, sometimes called a spectrophotometer or fluorescence spectrophotometer. Only a few compounds can fluorescent, and of those that do, not all photons absorbed will be converted to the fluorescent light. Okay, it's limited to the compounds which able to fluorescence. Otherwise, you cannot use the fluorescence spectrophotometer. Okay, now as you see on these slides, light source, okay, and then the light will expose onto the sample cuvette or transmit through the sample cuvette, and now only the solid which fluorescence, okay, will reflect the light, okay, and this reflected light is come through the filter onto the detector, and we measure the intensity of the fluorescent light which reflected, okay, via the fluorescent solid. Okay, this also show the mechanism of reflectance spectrophlorometer or spectrofluorescence meter. As you see, light is come through the monochromator, which exaggerated the intensity of the lights, and then through the test tubes, okay, and then if you have a molecules which fluorescence, they will reflect this light onto the detector, okay, and detector will measure the intensity of the lights. Okay, now, okay, any questions so far? Now I am going to discuss maybe a few more slides, then we will finish for today. But please feel free, ask me if you have any questions. Okay, each these instruments, the techniques are basic techniques, main techniques that we are using in clinical laboratory. They implemented into a huge instruments, and they are measuring billions of tests daily. And as you see here, the nephelometry is other kind of the techniques that we are using. Nephelometry is the measurement of light that has been scattered. Okay, it is different from reflectance, from absorbance or transmittance. Okay, turbidimetry is the measurements of a loss in the intensity of the light transmitted through a solution because of the light being scattered. Okay, this turbidimetry also a kind of the nephelometry, but these are different from each other. In nephelometry, the light will the scattered light will measures. Okay, in the turbidimetry, okay, the light scattered light also will measure, but it is due to the turbidity. Okay, the solution becomes turbided, and then we'll be able to scatter the lights. Okay, turbidimetry will measure the light that is scattered, okay, not absorbed or reflected by the particles in a suspension. Okay, nephelometry is used to detect the amount of the light scattered from the complex or compounds. And I am going to explain what does complex or compound means, how we are using nephelometry to measure antibody-antigen complex, the amount of antigen-antibody complex. Okay, now you see here, okay, the technique that working in the nephelometry, laser or other high intensity light sources is used in the nephelometry, and then you send the light, okay, via the lens onto the test tube, and it will scatter, okay, via 70 or 90 degree, okay, angles, and then come onto the other lens. Okay, through the lens is the scattered light, okay, and then reach out on the detector. The detector is measure the amount of the light which scattered, okay, from the compound, from the complex, okay, antigen antibody complex in the test tube, okay. Okay, the principle of use for the nephelometry, okay, when the specific antigen coded latex particles are agglutinated by their corresponding antibody, we know that usually antigens are soluble in the bloods or other body fluids. Okay, when they make a complex where they combine with their antibodies, we know that for each antigen in the body will produce a specific antibody, which is specific to a certain antigen. Okay, if you want to measure a amount of the antigen or antibody in the bloods, patient bloods, and then you will use a antibody for antigen, okay, or antigen for antibody in the test tube. You put a antigen in the test tube. If there is an antibody, they will make a compound complex, okay, and then precipitate, okay, which help, okay, and for the nephelometry, okay, the light we scatter of the solution, okay, can be measured by the nephelometry, okay, because the light will scatter from these macromolecules, come onto the detector, and then we will be able to measure the amount of the light that's scattered. Okay? The formation of macromolecule complex is prerequisite for nephelometry and protein quantitation. Okay, if there is no antibody to antigen, or there is no antigen to antibody, that means okay, the macromolecule complex, okay, won't be developed, and then you don't I mean, you cannot get enough scattered light to measure the amount of the molecules desired. Okay, in order to measure via nephelometry, should be developed a macromolecules, okay, a complex between antigen and antibodies. Okay, this will be my last slide for today. And there are some advantages of nephelometry and disadvantages. As I said, a cutting edge instruments, nephelometry, and infrared high performance light emitting diode light source is used in these instruments. The advantage of nephelometry is an automated system that working rapidly. It is a reproducible, relatively simple to operate, and very common in high volume laboratories. You you will be able to measure 100 tests per hours. Okay, it has also many applications for cardiac marker, for serum proteins, for antibodies in hematology or in microbiology. Okay. However, it has some disadvantage, and it include a high initial equipment cost. Okay, and also interfering substances may cause protein denaturation and erroneous test results. Okay, the most maybe disadvantage of these instruments, this technology is high cost. Okay, now let's finish here for today.